All right, welcome. My name is Caleb Bailey. I was asked by a family friend, actually, to provide a little bit more detail about the COVID-19 vaccinations and why they may or may not work. Is it worthwhile? Um, I don't want to provide too much of my background. I'm not trying to sell myself. I'm not trying to sell this story. I do have a PhD from the University of Iowa in molecular medicine, and and so I understand the concepts, but I do want to reiterate once again, I'm not trying to convince anyone one way or another on what decision to make. That said, I have found in my life and as I observe those around me, ignorance often leads to poor choices. And when people have understanding of how things work and maybe even a bigger picture of what's going on, then 9 times out of 10, people will make the right choice for themselves. And that's what I want to do. I just want to present information so that anybody who watches this will at least feel empowered by the knowledge that they have of what these vaccines are, how they work, how the body's responding, all of the physiology behind that. Now, that said, I recognize the biggest challenge that I have is to not dive too far into this. I could talk for hours about this because it fascinates me and I love it, but I also want to keep your attention. So it's going to be oversimplified, and in that way, um, I just hope that I can convey the message that I want in a way that you understand. So I've titled this little presentation Adaptive Immunity, B cells, T cells, and the COVID-19 vaccines. And we need to talk about all of those in order to really understand what's going on, because there's still a lot of misinformation and a lack of understanding of what adaptive immunity is, why I should care about B cells, the cells that make antibodies, or T cells, these killer cells that go around and actually kill infected cells, and especially how these new COVID-19 vaccines work. Whether we're talking about the Pfizer vaccine, the Moderna vaccine, these are fairly new technologies using mRNA transfer and liposomes. It's cool stuff, and I want to be able to try and address all of these in a simplistic sort of way. So the first thing that I think I want to talk about is simply I want to answer this question. What is a virus? At its most fundamental structure, a virus is simply a bag of protein, or a bag made out of protein. And within that bag, you find some sort of genetic material. Now, genetic material, what are we talking about here? We're talking about it could be DNA, it could be RNA, it could be double-stranded, it could be single-stranded, it could be positive sense, it could be antisense. There's lots of different viruses, and they have different strategies. But essentially, that's all it is. It's a protein coat, and within that protein, you've got some sort of genetic material. Another question, and even a better question, is why do viruses exist? What are they meant to do? That is a debate for maybe another day, but they're really good at transferring genetic material. And especially if you look at bacteria, because they don't reproduce by sexual reproduction, they lose genetic diversity. They just essentially replicate by making clones of themselves. And so one prevailing theory is that viruses evolved alongside bacteria to help transfer genetic material from one bacteria to another in order to increase genetic diversity. So that's kind of, coming from that Evo-Devo perspective, that gives us a good understanding of what viruses are good at doing. They're designed to infect and transfer genetic material. The coronavirus, this is a family, Coronaviridae. Typically not a lot of these viruses infect humans. There are a few, and usually you get these as children. This is why for about 30% of the population, They have some inherent adaptive immunity built into them because they were sick from another coronavirus a long time ago, and that's kind of stayed with them. The virus itself, coronavirus, is a spherical virus. The diameter of this is about 125 nanometers. It kind of gives you an idea of the size of it. And what makes these things unique, what gives them their name, are these spike proteins. And this crown or this halo around the virus, as you view it through an electron microscope, is what gives it the name coronavirus. And really, there are 4 proteins. There's the spike protein, there's the membrane protein, there's the envelope protein, and then there's this nucleocapsid. We're not going to delve into all of that. Really, what I just want to focus on is this spike protein. Because this is how the virus infects cells. For every virus out there, we kind of have to follow a rudimentary 5-step process for getting and infecting cells. And we can list those real quick. First of all, we have to be able to attach. This is critical, right? Different viruses infect different cells simply because they attach to those cells. If they can't attach to a cell, they can't do anything to get in. This is why different viruses give you different diseases. Ebola, right? It's a hemorrhagic fever. That's a nasty virus. It infects endothelial cells. That's not what coronaviruses do. These infect respiratory epithelial cells. Why? Because they can attach to them. So we have attach, and then we have entry. They have to get into the cell specifically, and the only thing that really matters is that genetic material that we were talking about for the coronavirus, it's single-stranded RNA, that has to get into the cell. And then once that's in the cell, they co-opt all the cellular machinery, everything that the cell's doing, this big factory. They say, no, we're going to turn that factory into a virus-producing factory. And so the next step is synthesis. And this is an important point to make. The virus itself doesn't have the capability to synthesize anything. It's just this bag of protein with some genetic material inside. It relies on the cell, once it gets inside, to make all the stuff that it needs to make for more virus. So we have synthesis of new virus parts. Specifically, we have to make more of the genetic material, in this case the RNA, and we have to make more of these proteins, the spike protein, the envelope protein, stuff like that. So the cell starts making all this stuff. And then once we make it, it gets assembled. And then that leads to some sort of release. And similar to many other infectious human viruses or mammalian viruses, this is an enveloped virus. In other words, what it carries with it is actually part of the cellular envelope from the cell that it came from. That's a little bit complicated, but that's what we're looking at. It's a single-stranded RNA enveloped virus. And it's this spike protein that we're super interested in because this is what it uses to attach, step number 1, and enter into the cell, step number 2, so that it can start doing its thing. So inside we have this RNA, and that's essentially this virus. Now, to help us understand how a vaccine works, let's first understand what a viral infection looks like. All right, so I'm a lousy artist. You can judge me if you want, but this is the point, right? This virus is spread by aerosols. Maybe you've heard of aerosols, nano droplets, whatever it is. There are different sizes these come through, but there are these particles of water that come out of people's mouths when they breathe, right? Anybody can do this. You walk up to a mirror and you breathe on it, and it gets kind of cloudy and foggy. That's because there's moisture coming out of your mouth. This has to do with vapor pressure of water. It has to do with lots of things. The point is, the virus is actually hanging out inside of these droplets. So we zoom in, we see, we see this droplet, and this is loaded with these little viruses. This is why masks are so important. There's big debate on, how effective masks are at helping you. They're not designed to help you, the wearer, right? That's not the point. The point is, as you breathe out, you're filtering these viruses so that you're not spewing virus into the environment. And that's the effectiveness of masks. And it is a very, very effective strategy. Does it protect you, the wearer? No, it protects those around you and is a great way to try and help those around you, especially those more susceptible to this. But anyway, so here's this virus. It's in this particle, and in it goes, up this guy's nose or in his mouth. And let's come in and let's zoom in and let's look at some cells right in here and see how this might work. So we got this basement membrane. We got some epithelial cells. They're held together by tight junctions so that nothing can get through. And along comes our virus. And remember, it's got these spike proteins on it. And it's going to attach, and there's still a little bit of debate, and we don't understand the full story, but we do know that a key receptor that's going to grab onto this spike protein is called ACE2. Stands for angiotensin converting enzyme, ACE. We don't need to delve into exactly what this protein does, but it's hanging out up here, and it's going to grab onto this virus. And through this interaction with ACE2, there's some cleavage events, there's some other things, and boom, this thing gets sucked inside the cell. And really, remember, what we're interested in is that piece of RNA. Because RNA, like DNA, but slightly different, contains a code, a message. It's just made up of A's and kind of, like that movie Gattaca. But we have A's. Instead of T's, we have uracil. So we have U's and we have G's and we have C's. It's just this code that is designed to tell the cell how to make a specific protein. So once this comes in, this is the advantage of the mRNA. It doesn't need to go to the nucleus. In this case, this virus doesn't integrate into the nucleus. It's not DNA. It's not even a retrovirus. It doesn't do any of those things like that herpes would do, for example. It's already got this RNA, and the RNA, remember, this is just a code to know how to make protein. And remember, we talked about the proteins it's going to make. It's going to make spike proteins, membrane proteins, envelope protein, nucleocapsid proteins. That's about it. And all these things are going to assemble and make more virus. And then, so as we just talked about this, right? We've got attachment, we've got entry, and now we've got a ribosome that is going to read this RNA code, and this is going to give us synthesis. And then, of course, we're going to get assembly and release of more virus. So one virus goes in, thousands, maybe even millions of virus come out. So out they pop. There's one. And now there's lots. Because the one virus went in and turned this cell into a virus-producing factory. Now, how would your body fight something like this, right? The end result is we got to kill this cell. It's not just going to stop making virus. It's going to keep doing that until it dies. And my job is to kill it fast. And that's where the other cells that we talked about come in, T cells in particular. So let's say that this neighboring cell is also infected. How does the body know it's infected, and how is the body going to respond to it? Well, to answer that question, and we'll come back to this cell in just a second, but to answer that question, let's just give a brief review or overview of your body's immune system. roughly speaking, we can divide this into two different sections, what's called innate immunity and what's called adaptive immunity. Innate immunity, we would consider things like, you strike a fever. That's going to help T cells fight a little bit better. It's going to do things to help your immune system. Things like phagocytes. These are nonspecific or arguably semi-specific cells that just like to chew up stuff. So we'll just drop Pac-Man. These, you have a limited, there are only a few types of phagocytes in the body, but they're super important in essentially cleaning up trash. And that's what they do. They chew on stuff. Things like complement proteins. This might be foreign to you, but within your blood, you have several types of proteins that work together to help an immune response. They can recruit through chemotaxis. They can bind to, and the word is called opsonize. It's essentially coat something to make it look tastier for phagocytes to come and get it, kind of like putting ketchup and mustard on a hot dog, right? It makes it actually taste almost good. And so the phagocytes would come and get it. That's something that complement proteins would do. These are innate because I'm, for the most part, born with them, and I don't have to teach them anything. This stuff just happens. But adaptive is different, okay? Adaptive is highly specific, very lethal for whatever it's targeting, right? Cells, bacteria, whatever. But here's the key. It requires essentially a two-step process. So it requires, we'll just call these selection and activation. Now, I don't want to go into the details of how this works. That would take too much time. But recognize it is a two-step process, and that helps protect against autoimmunity. Now, autoimmunity happens, right? And that happens when the selection and activation process just gets a little bit off. And now all of a sudden I have these T cells that are targeting my melanocytes, for example, and then I get white patches on my skin or my lips or my gums, vitiligo. You could see this even in certain types of baldness where hair falls out in patches. Diabetes, certain types of diabetes where these cells target the pancreas and specifically kill the cells that make insulin. It's a class of type 1 diabetes. So autoimmunity happens, but there are a lot of things that the body does to kind of prevent that, right? And that's why we have this two selection process. I think this is where a lot of people get confused. When we think about the specificity, when we think about the lethality, what are we talking about here? We're talking about cytotoxic or killer T cells. These are, well, we'll just call them killer T cells. And also, we can have B cells. B cells produce antibodies. B cells don't directly kill. Antibodies don't directly kill. Antibodies just bind to things, but that can cause all sorts of problems too. So killer T cells, and we'll put B cells on this list as well. These cells have to be selected for and activated. And that's an important thing to understand. I'm not born with T cells that automatically know to fight COVID-19. I am born, and then throughout the first 5 or 6 years of my life, with a lot of T cells and B cells that are what we call naive. I will carry these naive T cells and B cells throughout my entire life. Most of them, they're circulating in the lymph, they're circulating in the blood, but most of them are hanging out in lymph nodes. You get a swollen lymph node. It's because the activation and selection process is happening for these cells. So I have all of these naive cells. Every single one of them, and this is fascinating. Every single one of them is different, specifically in their B cell receptor or their T cell receptor. And if you want to learn more about this, it's kind of a complicated process, but it's cool. It's a process called VDJ recombination, where these cells go in and there are certain parts of the DNA, they kind of mutate them and swap them around and change them up so that the DNA for this, that codes for the receptor is actually different, randomly different in every single one of these cells. And most of these naive cells will go throughout my entire life without ever being selected for and ever being activated. Now, they do have to they have to pass a couple of tests, right? We were talking about autoimmunity before. If a T cell recognizes self, that's a problem. I don't want my T cells attacking me. And so as they go through this early stage learning, they look for cells, the body looks for cells that have the propensity to recognize self. And if the cell does, the cell is killed. It's no longer allowed to progress. So this is one of the things the body can do to protect against autoimmunity. So now I've got all of these naive cells, and they're just waiting. They're waiting for the opportunity to see what we call an antigen. An antigen is just something that is immunoreactive. It binds to antibodies, or it binds to these receptors, and it's going to stimulate them. So to understand these guys, because this really is at the heart of how our immune system works. Let's go back up and look at this cell. Something that most cells in the body do, they present antigen on what's called MHC class 1. That's a little bit complicated, but every cell in the body essentially has dog tags that are unique to you. Your MHC is different than everybody else's MHC in the world. It's your dog tags. It's your ID. This is why you get transplant rejection, for example, because the MHC molecules on the transplanted organ are not yours, and your body recognizes that. And so you have to take medication to kind of dumb down your immune system, and you become immunocompromised because of it. How does it work? Well, these cells are constantly chewing up things that are inside of them. And just imagine a cow chewing its cud. It's chewing, and then it regurgitates, right? You throw something up, you're like, look, this is what I was eating for breakfast. That's how these cells work. So remember, we've got this RNA in here, and it's being translated to protein. And so I've got this spike protein that's being made inside the cell, and it's being loaded into new virus like we talked about earlier. But as part of this process, the cell's always taking stuff and chewing it up and presenting it on MHC molecules. And so this is actually a little bit of it is going to be chewed up, and here's going to be my MHC right here, and it's going to be holding a bit of that spike protein. Now, if it wasn't infected, it's going to be holding a bit of normal protein, right? But the cell's like, I've seen that before, that's no big deal. But this one's foreign. And so that's hopefully, right, we find a match in all of these naive cells that we've been having. Hopefully we find a match in that. So along comes this killer T cell, also called a CD8 T cell for reasons that I'll show you. This is a killer T. And the first thing that it's going to do is it's going to reach out with one of its receptors, and it's going to shake hands with your dog tags, your MHC protein. That's how it knows that you are you and not somebody else. That's a good thing. So it extends its friendly handshake. As long as it receives a friendly handshake back, you're good. If you can't give that handshake back, now you've got a problem. The killer T cell is just going to kill you. But here we've got that MHC, it matches up, everything is great. Now we have the T cell receptor. And this is what we were talking about earlier, where every single one of your naive T cells, it has a different T cell receptor, randomly selected, randomly created, so that you can always find one that can bind to any antigen you can imagine. Well, now we've got an antigen. We've got this coronavirus antigen. And this killer T cell happens to recognize it. Remember, we talked about a two-step process: selection and activation. Here is the selection part. If this works, if we have this interaction right here, then this killer T cell, now all it needs is a second signal for activation. We don't want to go into the details of that, but it's going to get that through another type of T cell, a helper T cell. Here comes that second signal. And this is a form of cytokines, just little molecules that come and bind to it. And it activates this thing. So now we've got selection here and activation here. And this killer T cell, with its unique T cell receptor, has now been selected and activated. It's going to do two things. Okay? Once it's gotten selected and activated, it's going to make a clone army. So it's going to proliferate and it's going to make a bunch of itself. And these things are my warriors. These things are now going to go out, and anything that receptor identifies, which is going to be this spike protein on the coronavirus, this cell's going to kill it, right? It's going to kill this cell, right? We get out the samurai sword and we kill that cell. That's this virus-making factory. We get rid of it. It's okay. The body can make a new one. That's no big deal. We just got to get rid of this one. So the clone army is now out looking for cells infected with virus that are expressing these chewed up bits of spike protein on their MHC molecules, and it's going to kill them. The other thing that it's going to do is it's going to create a memory cell. And these aren't going to the front line. These are going to kind of be stored and set aside for another day. But this is the idea of adaptive immunity. I'm going to clear out everything that I've got going on right now, and I'm also going to store memory cells so that when I see this same antigen again, whether it's next month or next year or 5 years from now, 10 years from now, I've already got a T cell who has gone through the selection and activation process ready to go. I just pull him out of retirement, get him to do his job, and that's why when we're exposed to these things later on in life, we might not even know we're sick. We might show very mild symptoms, but we're a lot quicker in responding to it because we've already previously gone through the selection and activation process. Now, depending on the type of cell and all that stuff, and we're seeing this today, right? Do we need boosters? Yes, sometimes. You think about tetanus. You need a tetanus booster every 10 years. Just to kind of wake this system up. And, maybe it is just a few months. Maybe it's a few years. It just totally depends on the type of cell being produced and the antigen and the response, all the stuff that we've been talking about. But that's the premise for how this system works. Now, B cells would go through a very, very similar process. There are some nuances, there's some differences, but at the end of the day, it's very similar. The only difference is that a B cell is not a killer cell. The B cell is not going to kill cells. The B cell makes antibodies. It's going to make a clone army, all these plasma cells secreting this antibody that's specific for that thing. And what do these antibodies do? They bind to it. One of the things why these antibodies are so helpful is because they can neutralize the antigen. If we zoom in and we look at this, we've got antibodies coming in, they're being made, right? These antibodies can bind spike protein, and if all of my spike protein is covered with antibodies, guess what? These viruses can no longer bind to the receptor ACE2 on the surface of these cells and get in. I've just blocked the ability of this virus to bind to, attach, and enter cells. Plus, remember we talked about opsonization. That's what these antibodies do too. So these recruit phagocytic cells that will come start chewing this up and destroy these things. So there's a couple of different ways that antibodies work, but that's all they do. They bind and they neutralize, they opsonize, they agglutinate, all these different things just to help clear this substance. Okay, so now we kind of have a general sense of what a virus is and how the body would normally respond. Recognize that time is of the essence. When we think about people getting sick with COVID, if they've never been exposed to this virus before, the selection and activation process takes time. It could take 2 to 3 weeks to get the clone armies up and running so that we can start clearing the virus out of the system. And often people don't have that kind of time, especially people who are more susceptible. If you get a lot of this virus and it's infecting these respiratory epithelial cells, now you have a hard time breathing. That in and of itself, you kind of become immunocompromised. It slows this whole process down. Anyway, your body never has the time to go through the selection and activation process before you succumb to the disease, your inability to breathe or to ventilate because of this virus. So let's now talk about what a vaccine can do to help this process. So if we look at COVID vaccines, the two main types that we're seeing right now are the Pfizer Moderna vaccine. Those are very similar, and then the J&J vaccine. And they actually work very differently. But at the core, they're similar. So let's first look at the Pfizer vaccine or Moderna. These vaccines are mRNA vaccines. What that means is that they're very, very safe in what they do. Remember, the mRNA is simply a code that provides directions for making proteins. You know, your body, you're made up of 50,000 plus different kinds of proteins, whether it's a receptor or an enzyme. There's lots of different kinds. And your DNA contains the master code to know how to assemble these proteins, how to make them. The DNA is copied into what's called a messenger RNA or an mRNA, and then that mRNA is essentially translated. The code is read by a ribosome, and the ribosome assembles the protein according to that code. The mRNA is delivered in these little nano carriers, really called liposomes. This is an oversimplification, but just imagine essentially a tiny little ball made up of fats, phospholipids, that contains this piece of mRNA. And that mRNA only codes, in this case, for the spike protein. There's no way, right? This is one of the things. Am I going to get sick? Am I going to get coronavirus by taking this vaccine? No, it's literally impossible. Now, that's not the case for previous vaccines. You go back, to the early days of vaccines, take polio, for example. People got sick from taking the polio virus. It was attenuated virus. You were actually getting the virus injected into you. But they saw the effectiveness, people did, and they knew that that was much better than actually getting sick by polio. And so it was very worth it to them. That can't happen with these vaccines. It is literally impossible. All we're doing is providing the mRNA for a piece of one of the proteins that this virus contains. You can't make a protein from it. The liposome is designed to get into the cell. It's not super effective, and this is where you get some of the side effects of the Pfizer Moderna shots, but it's a decent way to do it, and it's very, very, very safe. So if we go back to our cell, this liposome comes along, and the liposome can get through the cell membrane. That's what it's designed to do. And lo and behold, what do we have? Now we have this piece of mRNA specifically designed to make the spike protein. And what's going to happen? This cell is going to start making spike protein. And pretty soon, what are you going to see? You're going to see this bits and pieces of spike protein expressed on MHC being presented by this cell. The key difference is this cell isn't making virus. It's just presenting the spike protein. We go through the same selection process, right? We've got our killer T. And he comes and identifies that first by shaking hands with CD8. There we go. We shake hands. And then we've got the T cell receptor that recognizes that. What have I just done? I have selected and activated the right killer T cell without having any of my cells infected with virus. The killer T cell is going to do the same thing that it normally does. It's going to make a clone army. It's going to go down and make some memory cells, right? It's doing all of that stuff. But it gets to the front lines. It's like, oh, there's nothing to fight. There are no cells infected with virus. But the cells are primed now and ready to go when that infection might come. Again, it's the same thing with B cells. The B cell is going to go through, because of the vaccine, it's going to go through this process of selection and activation, and it's going to start churning out antibodies. There's not a whole lot in the body for those antibodies to bind to, simply because you're not infected. But when coronavirus does come and try to infect, the B cells and T cells, the memory cells in particular, are primed and ready to go out and meet the infection head on. I know there's been a lot of worry. Well, I got vaccinated, but I still got sick. Well, yeah, that's not the point. It's not designed to prevent infection. It's designed to identify infection very, very, very early and clear it out of the system, so that most of the time you didn't know you were infected, or if you did figure it out, right? You tested positive or whatever, it's going to be very mild. Why? Because the selection and activation process was already done, and the clone armies have been built, and it's fighting long before you ever knew you were infected. Now, scroll down here. The Johnson & Johnson vaccine works just a little bit differently. The Johnson & Johnson vaccine is actually an adenovirus. Adenovirus is just another type of virus, right? We're talking about coronaviruses, retroviruses, adenovirus. It's just another type of virus. Adenoviruses can make you sick. They are associated with common colds, things like that, but not this adenovirus. Okay? Remember what I said at the beginning. When we think about viruses, it's just a bag of protein containing some sort of nucleic acid material. These adenoviruses have been altered in a laboratory. And before you freak out when I say that, what it means is we've gone and we've taken out all of the bad things from the genetic material. We know the code, we know the sequence, we know what all of these genes do. We say, well, let's just take this part out because this is the part that allows it to replicate. Let's take this part out because this isn't any good. Let's just put into this virus the very, very basics of what it needs to get into the cell and deliver the message. That's it. The cell doesn't really become infected. It's not going to make more virus because the code that virus is carrying doesn't allow it to. This is a very, very safe system, and it's been around for a long time. People play with these kind of viruses all the time. It really just isn't that big of a deal. So in comes this virus, and it delivers the mRNA for the spike protein. So the difference between the two vaccines is the mechanism of delivery. The J&J vaccine delivers with the adenovirus, again, which is very, very safe. And the other two, the Pfizer Moderna vaccines, deliver with a liposome. But at the core, we're still getting this mRNA sequence in here that is specific for the spike protein. The spike protein is translated, expressed, and used to select and activate my killer T cells and my B cells. Now, just to address a couple of things about some of the myths that you hear regarding these vaccines. The biggest one is, are they safe? It hasn't been enough time to really vet out whether they're safe or not. The truth is that there has probably not been another vaccine that's been vetted and tested as much as these have. The fact that it came about in just a few short months compared to years simply has to do with the fact that so many companies were focused on one common goal, and by working together, they were able to achieve something quite remarkable. But never has a vaccine been tested and vetted as much as these. Number two, is there vaccine injury possible? There's always the risk of vaccine injury. If it's the flu shot, if it's, your MMR, whatever it is, there's always the risk of vaccine injury. But those vaccine injuries run in the realm of 1 in a million to 1 in 5 million. And the odds are very much in your favor. You compare that with, for example, aspirin or Tylenol, and you look at some of the risk factors, cerebral hemorrhages, things like that, associated with these common drugs that we take all the time. They actually have much higher risk factors for them than these vaccines do. So across the board, as far as medications go, when you can find a medication that is 1 in the million for bad things happening, that is just incredibly safe, and we jump all over, right? That's a great, great thing. Another one that I've seen is affecting women's health and sterilization. There was a false report that had no basis that came out a little while ago, talking about how this spike protein, when antibodies are made against the spike protein, that during placentation, there's a specific protein made in the placenta that's very similar to the spike protein. And so if you have antibodies against the spike protein, then these antibodies would also attack the placenta. The answer to that is absolutely not. There's no similarity between placental proteins and this viral spike protein. Again, this goes back to the selection process that we talked about earlier at the beginning. The body first makes sure that these naive T cells and B cells don't recognize self, any part of self. And so in that way, they're incredibly safe. Another thing that I've heard, people want to say, well, I'm going to wait a few more years because I want to make sure that they're safe. Honestly, that's kind of pointless. We have never, ever seen any vaccine injury take place longer than 3 months after the vaccine is given. So if something bad is going to happen associated with that vaccine, we will see it in those first 3 months after the vaccine's been given. And so we don't need to wait years to know that this is safe. So all of the people, all the trials, everything that's gone into this, it's been very safe, it's been very effective, and the vetting process has been very professional and secure. So as far as those things go, there's nothing to worry about. We showed you with the technology why there's really no way that you could actually get coronavirus from this vaccine. It's completely impossible, simply because you're not getting infected with the virus. You're just tricking the body to make a certain part of the virus, and then the body knows how to respond to that thing. the side effects, especially of the Moderna and Pfizer vaccines, are typical of exposure to mRNA, right? The body, going back to the innate immune system, the body is very sensitive to random pieces of mRNA just floating around. And so it will induce fever and some of these other symptoms, body aches, things like that, simply because that's what the innate immune system does when it sees RNA. The body's very sensitive to that. So those are predictable side effects that, it's nice if you take something and not have any side effects, but for a day, you're going to be sore, right? The shot in the arm is going to be sore. There's a good chance that you're going to have a fever from it for a few hours. That's a typical response that the body is making just to random RNA. And we could talk about toll-like receptors and all the stuff going on there. That's another topic. Efficacy. I don't know, and I'm not the expert here, but I don't know that there has ever been a vaccine shown to be more effective than the 90+% efficacy that we saw with the Moderna and Pfizer vaccines. And then I remember when the J&J vaccine came out and it was like down in the 80s. They're like, oh, it's much worse. No, that is still so high, right? I mean, your average flu shot that comes out every year, sometimes those efficacy rates are, 10%, 20%, 30%. It kind of depends from year to year, but to have numbers as high as they were with these is really an amazing thing. So from a safety point of view, there's no way you can get sick. It's a very effective way to help the body go through the selection and activation process to prime the T cells and the B cells so that when you are infected with an actual viral infection, those cells are ready to clear it out quickly. Is it going to make it so you don't get sick? No, that's not what vaccines are designed to do. They're designed to prime the body to fight quickly when that infection comes. This is why people still need to think about wearing masks, even if they've been vaccinated. I've been vaccinated, but if I am exposed to that, I might not even know that I'm sick. And we see this in the news, right? People, especially in the sporting field where testing is so common, somebody shows that they are positive for coronavirus. They don't feel sick or maybe the symptoms are very, very light. That just shows that the vaccine is doing its job in fighting the infection that's come. But importantly, that person is still infected with a virus and could still potentially transmit that virus to others through the aerosols in their mouth. That's why the CDC is pushing right now for even vaccinated people to wear masks just to help curb that process. So hopefully that wasn't too much. Hopefully you have a better understanding now of how the body's immune system works, how a vaccine is designed to work, and at least we've covered some of the basics of the differences between the Johnson & Johnson vaccine and the Pfizer Moderna vaccines and how they do their job. Is it probable that we're going to need booster shots for these? Time will tell. Research is suggesting that, yeah, that's the case. But remember, we get booster shots for lots of different things. That's just, it's not that big of a deal. It's just part of the process. So by far, historically, if we look at the last 50 to 60 years, the best way to curb viral infections is to prevent them. And that's what vaccines are designed to do. But in that word prevention, please recognize it's still possible that you could get sick, but you're probably not even going to know you're sick. Your body's going to be able to respond very quickly. And if you do get sick, the symptoms are going to be very mild. That's just how the body works. So hopefully that helps. Good luck, and feel free to share. And with this knowledge and understanding, now you're ready to make whatever decision you need to make.